Hello and welcome to Basic Biology, and today we're doing the structure and function of the different organelles that you find within a cell. So I'm just going to start by drawing a smiley face, and this organelle's function is basically to process and package proteins into vesicles for either movement to the plasma membrane for insertion or for release out of the cell. This is called the Golgi apparatus. So, how is that protein made in the first place? Well, if we move backwards, we can see there is a rough endoplasmic reticulum. It is called the rough endoplasmic reticulum because it has little bobbly bits on the outside, and those little bobbles are actually called ribosomes. So it is studded with ribosomes, making it rough. And this is where protein synthesis can occur. So it's safer to have your proteins being synthesized inside these cisternae or sacs of the rough endoplasmic reticulum so that it is protected from any chemicals that might be out here in the cytoplasm. So the protein will be synthesized by the ribosomes and travel through the rough endoplasmic reticulum, then be packaged into a vesicle where it will then be moved to the Golgi apparatus. So this is the rough endoplasmic reticulum. So where do the instructions come from in the first place? Well, over here we have a nucleus which has a double membrane. It has small pores in the nuclear envelope, and that allows small molecules like mRNA, which have been synthesized inside the nucleus, to leave, and those reach the ribosomes, and the instructions are then followed by the ribosomes to make the protein. We also have in here a nucleolus. So this nucleolus is the site of RNA synthesis, and in particular rRNA, which is basically where the ribosomes are made. So the nucleolus makes ribosomes. Here in the nucleus, this is where all your DNA will be found. And it's from that the copies are taken and used to make mRNA. So you have a copy of the DNA made, so this little section here, which will code for a particular protein, will be copied using RNA to make mRNA, and that will leave through the nuclear pore. The DNA cannot leave because it is just too long a molecule to be able to get out through that nuclear pore. So far then, we've talked about the nucleus, the nucleolus, the rough endoplasmic reticulum, the Golgi apparatus, and we've got vesicles over here which are doing the transporting. We've also got in this cell a mitochondrion, which is the site of aerobic respiration. So it has these little infoldings of membrane, so it has two membranes, one on the outside and one highly folded on the inside. The inside one, these are called the cristae, which is plural, and these are the mitochondria, or mitochondrion if there is only one, and in your exam if it points to just one mitochondrion, call it a mitochondrion. The cristae contain enzymes for aerobic respiration. It is absolutely essential that when you now mention respiration, you decide whether it is aerobic or anaerobic. If it is taking place in the mitochondrion, it is aerobic respiration. Alright, so what other organelles have we got? Well, we also need to synthesize lipids, and synthesis of lipids occurs within the smooth endoplasmic reticulum. So it's a very similar structure to the rough, but as the name implies, there are no ribosomes present, and that's where we get the synthesis of lipids. So we've got smooth endoplasmic reticulum, mitochondrion, vesicles, Golgi apparatus, rough endoplasmic reticulum, nucleus and nucleolus. If we're talking about plant cells, then we'll find chloroplasts. And just like the mitochondria, these also have two membranes, one on the outside and one on the inside, which has been specialised and forms something called thylakoids. And basically, thylakoids are just lots of the inner membrane folded around to make sure that you have the biggest surface area possible for light capture. So that's obviously needed for photosynthesis. So this is a chloroplast. Okay. In the cell, sometimes we're talking about moving around these vesicles. Well, to move them around, you're going to need a cytoskeleton, which is basically a series of lots of tubules. So we've got tubulin, microtubulin, and microfilaments like actin. And what these guys do is these make it possible for these vesicles to move around. They also provide the structure and the shape of the cell in the form of the cytoskeleton. So we have the cytoskeleton for the movement of vesicles, for the movement of organelles within the cell, also for the maintenance of the structure and the shape of the cell, and also in white blood cells for the movement of the cell membrane itself. So if we have a little drawing down here, this is a white blood cell and this is a little bacterium. You need this white blood cell to engulf. In order to move these parts of the membrane so that they can surround this bacterium, you need cytoskeleton. And what will happen is that white blood cell will start to surround, and eventually those two parts of the membrane there will fuse, and you'll end up with the bacterium surrounded by a phagosome, and the cell is now complete again, but with the bacterium inside and contained. So this is compartmentalization within this phagosome, which is very handy. And then you can have something called a lysosome, which contains digestive enzymes fusing with that phagosome so that you get a phagolysosome, and then this bacterium would then become broken down. The cytoskeleton is also important for the movement of the cell. So if we had, for example, something like a paramecium, so that's a eukaryotic cell with a flagellum or underlipodium, then it is able to propel the cell forward. Again, that will need lots of mitochondria to carry out aerobic respiration to provide plenty of ATP to power this cytoskeleton. So anywhere where you see movement being mentioned, remember you'll not only need the cytoskeleton, but you'll also need mitochondrion to provide your ATP. Hope that helped. Good luck.